Okay, so good afternoon to everyone. We have today the 12th webinar of Translator, and we have the pleasure to have two speakers from the Karolinska Institute, Vicent Pelechano and Yujie Zhang. I think Vicent is a PI in the SciLifeLab in the Karolinska in Sweden. And he will make an introduction of all the things that they are nice things they are doing in his lab. And later, then we will have a talk from Yujie, who is a postdoc in Vicent's lab. So if you want, Vicent, please. Thanks
a lot. Thanks a lot for the invitation and for the introduction. So as Paula say, I'm going to briefly talk about what we do in the lab, and I'm going to focus on co-translational RNA decay, so on the translation-related work that we do in the lab. So our lab has quite a diverse interest. We are interested in RNA biology, but we kind of has a broad interest from chromatin organization and transcription to the decay of the RNA. We have three main topics in the lab, one focusing more on single-cell epigenetic heterogeneity and cell plasticity. A second one focused on the crosstalk between ribosome dynamics and RNA decay. That is what I'm going to talk mainly. And another angle where we try to use our fundamental knowledge in these different areas to try to bring them a bit closer to the clinic, because we are a medical university. And I will also briefly mention now. But I'm going to focus mainly on the work in the crosstalk between translation and RNA decay, as this is the Translator meeting. So I want to just to start regarding why we should care about, why we care in our lab about RNA decay intermediates. Normally, when you look for the central dogma of gene expression or molecular biology, you have DNA, RNA, and proteins, and you have transcription and translation. And of course, at some moment, the RNA and the proteins have to be recycled and so on. But we, many years ago, discovered in a serendipitous way that, in fact, the RNAs that are being degraded, there is information that we could leverage. And if you look to all the RNAs in the cell associated to ribosomes, you have the bulk of ribosomes, let's say, that are producing proteins or store or whatever. And there is a fraction of them that are being degraded at the same time. So the degradation machinery is kind of degrading, but the ribosomes are still there. And if you analyze, in this case, specifically the 5' phosphate molecules that are changed here, this is our exonuclease, and then you map them to the genome, you get kind of a pattern of degradation. And one thing that we discovered at that time is that if you align all these 5' phosphate reads, for example, for the stop codon, you get this pattern, this periodicity every 3 nucleotides. So the RNA degradation is not totally random, but there is a periodicity every 3 nucleotides. And if you focus here in the stop codon, you can see a huge peak. Long story short, we saw that the exonucleases present in the cell are trimming the mRNA co-translationally in a way that make an in vivo footprint or toe print, because it's only from one side of the ribosomes, and we're leveraging that information. This data is from Saccharomyces cerevisiae. I will talk later about other organisms. But basically, we saw this kind of very clear association. So somehow in the RNA degradation, we can get information regarding what's happening at translation level. Just to show a bit more clear example, here we decided to align all the reads. This is a metagene level for the histidine codons that is positioned here in 0. And we grow the cells in SD, that is a synthetic defined media. When we grow the media in a media depleted with histidine, we are forcing cells to do their own histidine. And that leads to a stalling of the ribosome because there is not enough histidine. And this stalling of the ribosomes makes that there is an accumulation of degradation intermediates here because the ribosomes is slower and is trimmed. And when we really block the histidine biosynthesis with an inhibitor, we see that this stall is so big that you have multiple chains of ribosomes. So I put a bit more time to explain here because both myself and then Yujie will show this kind of plots a few times just to tell what it is. So this is basically mapping the 5' phosphate molecules in a method that we call 5p-Seq. So we have worked a lot both in the computational and experimental analysis. So we have a easy to analyze and visualize analysis pipeline. This one developed by Lilith, a former postdoc in the lab, and allows to basically automate this process for analysis and make it easier to interpret. And also Yujie before develop simplified methods to do this also easier in the lab. I'm going to just sideways and talk about other organisms in this case, just to give a different angle. And then I will let Yujie to focus on her project. So one thing that came here is that we were doing this in Saccharomyces cerevisiae, but we wondered is how in general is this in evolution? We knew that this was happening in cerevisiae and pombi. We know it's happening in many other species, in Candida, in plants. In humans, it's complicated. I can tell if anyone is. So it's not happening exactly this because there is other exonucleases, so it's not so clean. But we wonder, what about the prokaryotes? What's happening in bacteria? So this method is drag and simple because you don't do the experiment, the cells do it for you. You can apply it for a sample that has been stored in the fridge for years because the experiment was done already when you collected the samples. We know it's evolutionarily conserved in many eukaryotes, and we get a proxy for ribosome dynamics, even if we focus on the RNA that is being co-translationally decayed, and then we get a very long footprint. So having a long footprint and being applicable easy to any sample suggested us that maybe we could apply this to bacteria, especially to microbiome samples, like very complex samples that we collect from the environment or for patients that have many, many species. If we have a very long read, we can distinguish from which species is coming. And if we don't need to do the experiment, it's even better because it has been done already. So I put this like this just to make sure that we are not doing ribosome profiling. We have an information that has something to do with ribosome profiling, but we are not doing ribosome profiling, just to be clear. So we are good for ribosome dynamics, but focusing on these ones associated to decay. And this, as you will see later in Yujie's talk, has some advantages and some disadvantages. So when we go to bacteria, we realized that, in fact, it was more complicated than we thought. And there is not a direct homologue for XRN1, that is this X nuclease here in cerevisiae. But in some species, like B. subtilis, you have RNase that functionally does something similar. But for example, in E. coli, that's not present. So we did this with Susan and Lilith in the lab. This is B. subtilis, and we could see something very similar. We saw the pattern in the start codon and in the stop codon, and this is the 3-nucleotide periodicity. Then when we add chloramphenicol, we can see some alterations, and when we fragment in vitro the molecules and sequence them with the same protocol, we see that this is lost, just to show that this is biological. So basically, this exists also in many species, around 60% of the species of all bacteria at different degrees. And this is already published almost 2 years. And we can use this for many things. Just to highlight a couple of things is we use cells from bacteria and then added antibiotic, in this case, mupirocin, targets tRNA synthetase. And we saw that in 5 minutes, we could detect by sequencing the RNA degradation intermediates the impact of those in the cell. So we could get information of the phenotypic consequence of the antibiotic treatment very fast. And we can apply to complex samples. So here we mixed different species that were relatively L-Reuteri and L-Plantarum, and we could see that we could distinguish them, even if the species that we were focusing was less than 0.01% in the population. So we can really just have a complex sample at this angle. We have done many, many species. So in this paper, we published 96 species across 58 genera. We did feces, vaginal swab, soil. So it's a quite dense paper. But now we are following this in different angles. We are interested in the fundamental dissection of co-translational decay. And Yujie will talk about this later, so I will jump. But then we have some work working on trying to bring this to single cell and communities and develop molecular results for phenotypes. One thing that I just want to mention at the end, just before passing the talk to Yujie, is that we are trying to use this phenomena, especially this, to try to accelerate the detection if an antibiotic is going to work or not in a bacteria. Because using this, we think that using this kind of readouts, we can accelerate it. And as you have seen from a couple of slides, I have this conflict of interest because we spin off a company from the lab that is trying to bring this to the clinic. So that's co-founded with Susan and Lily, that were the first authors of this paper. So how people prescribe a correct antimicrobial? Of course, there is many guidelines, but if you want to know if an antimicrobial is going to work, there is two classical ways. One is the phenotypic test. So you test the effect of the antibiotic. Like, for example, this is the classical drug disk diffusion assay, that you put the bacteria and then you see, okay, these bacteria are inhibited while these antibiotics are not inhibiting this strain. And this is cheap, works, but it's a bit slow in some cases, and some bacteria don't grow very well, so that could be a problem. But this is the standard. And then you have molecular tests that you know specific genes or characteristics that give you a signature that tells you, okay, this has a resistance, and you can do a PCR or sequencing or MALDI-TOF or something like that to classify. These are normally faster, but they don't tell you necessarily the effect. And the other ones tell you the effect of the drug, but then don't necessarily, they're maybe not as fast. So we wanted to do something in between. So we wanted to do something molecular, but tell us about the effect. So can we measure the ability of cells to grow at a molecular level? And to do that, we are measuring the effect in the ribosomes, because of course you need the ribosomes to grow. So we are trying to use this as a molecular readout. Instead of trying to see cell divisions, we just see the effect in the whole team, and then take any questions now or later, and then pass to colleague.
Thank you very much, Vicent. Usually we have the questions later after the two talks, if you
That's
think
perfect.
okay. But I want just to say that this is a great example of how basic science can become applied and useful for clinical, interest. So I think it's really great the work you are doing. So if we have questions at the end, then we can pass now to Yujie Zhang seminar. So please, when you want.
Sorry. Can you see my screen now? Yes. Okay. So
Yes.
I will start. Yeah. So my name is Yujie. I'm now a postdoc from Vicent Pelechano lab. So today I want to show you the study I worked before with the title is Ribosome Modulate Transcriptome Abundance through Generalized Frameshift and Out-of-Frame mRNA decay. So I think Vicent already have a very nice overview of what is 5p-seq and and how to read the plot and how the differences of 5p-seq with ribosome profiling. So I will skip this part. I will directly go to the study of my topics. So basically, you will see here. So when we perform 5p-seq in yeast, so here we show one example in YPD, which is a very rich medium. So you will see this canonical pattern, which at -17 peak, which means that the ribosome is sitting at the A site. So very surprising very surprising when we perform the 5p-seq in CSM. It's a kind of a nutrition, very limited conditions. We see the -17 peak, it's shifted to -18 peak at the metacodon levels. So here I show you one example with proline codons, but we find that this massive shift is not only at proline codons, but all the other codons. So those I will show you later. So at globally, when we try to sum up all the frames in rich media, you will see the frame 1 is predominant. However, when cells grow in the low nutritions, this in-frame change towards to F0 frames. So this suggests that there is a massive frame shift happening in these low nutrition conditions. So we wondered if this happens with the subpopulation genes or globally. So what we do here is we define the frameshift index as the frame in frame 1 versus out of frame 0. In the condition of rich media, you will see on the x-axis, and also compared with the frameshift index in the low nutrition conditions. So in the y-axis, so each dot represents a gene. So we will see like there is at least 1/3 of the transcriptome undergoing this ribosome frameshift. So next, we wonder if this is also dependent on the stress. So what we did is we performed in the YPD or in the CSM, coupled with all the different kind of stress that may affect the translation process. However, we see like the nutrition. So you will see all the frameshift happening in the low nutrition condition, no matter of the stress. So means that nutrition is a main driver for this frameshift. So we also performed this experiment using the ribosome profiling, where we see the frameshift happening at a low degree than the 5p-seq. So from here, we are trying to propose our model. If you're thinking like under the low nutrition conditions, we will see all the ribosomes attach ribosome on those transcript. So they are following the slow decay path. So then using 5p-seq, we will detect those degradation intermediates as inframes. However, in the low nutritions, we saw this massive frame shift that could introduce premature termination codons. And therefore, they recruited NMD decay factors that fast degraded those transcript that are that producing this auto frame degradation intermediates. So to test this model and to test if NMD is involved this way in this pathway, so we use an independent method that can measure the RNA degradation rate. So here we are we are using the SLAM-seq, which can we will label the transcript and then differentiate if this is old or new. So we can measure the RNA decay rate in the two conditions. So in this case, we are doing the SLAM-seq in the YP and the CSM in the pulse and chase manner. So we will see the CSM degrading in the CSM conditions. RNA degradation is faster. So to test it, if the NMD is in this pathway, so we performed the SLAM-seq in the YPD and CSM in the wild type and also in the NMD, one of the NMD main factors, UPF1. So we saw the mutant and the wild type in the rich conditions. The RNA degradation, it's not so much change. But if we are looking at in the low nutritions, so if we knock out the UPF1, the degradation is much slower. So this suggests that in these low nutritions, NMD is actively degrades a chunk of transcriptome. So we also perform this using the proteomics to see how this long-term amino acid limitation reveals the whole cellular proteome under the conditions. So next we wondered, so what is the mechanism that causes massive frame shift and then RNA degradation? So there are two hypotheses that could induce this ribosome frame shift. So one is slippery sequence, that is a canonical ways for inducing ribosome frame shift. So when we checked with those we checked with those slippery sequence, we find there is no big change in the no big change in our conditions. And then we are thinking about if there is a hungry codon that can cause this frame shift. So what is a hungry codons? So we know that during the translation, so the tRNA ribosome will decoding the A site. So if the A site had limited charged tRNA or for some reasons, then they will somehow moved maybe 1 or 2 frames upstreams, and then finding the new codons, and then reinitiate the translation process. So we want to look at if this hungry codons induce this frame shift. So that we wanted to measure the global level of the codon usage. So that is remind us of this codon optimality, which measures the the balance between the tRNA availability and this codon usage. So for that we are define the frame shift index for each codons. As you will see, these 2 columns. In the first column, it's in YPD and then in CSM. So we ranked the frame shift index for each codons from high to low. We find high frame shift codons are more enriched in the non-optimal codons. So which suggests that the low codon optimality, instead of the program frame shift site, they are the key that induce frame shift in our low nutrition conditions. So we test this hypothesis. So all those experiments we have done, it's in yeast. So which shows that this mechanism is in yeast. But we also want to know if this is also conserved in bacteria or in humans. So this I will show you 1 slide later. But before that, I just want to show you that we are also using an independent approach. So to see if this frame shift is true under low nutrition conditions. So what we are doing here is like we have a reporter system where we have RFP and GFP, and in between we have the sequences we want to test it that could potentially induce a frame shift. And as a control, we also use a program frame shift site from HIV from the literature to see if this construct can properly detect the frame shift level. So as we can see from this, like in the YPD conditions, we will see the program frame shift site from the literature give us around 13% of the frame shift level. So just to mention that how we measure the frame shift level is like if there is a low signal of the GFP relatively to RFP, the high frame shift it is. So when we perform this experiment in CSM, we see there is a 50% reduction of the GFP relatively to RFP signal, which suggests this massive frame shift happening in these low nutrition conditions. Here I show you all CSM. And we want to see if we add some amino acid specific amino acid, can we rescue partially of this phenotype, like to decrease the frame shift. So here we selected some amino acid, and we show that they can partially rescue the frame shift phenotypes. And so then just come back. We show that happens in yeast. So we also show that this autoframe translation decay it's also conserved in bacteria. So here I show you in 2 bacteria example. One is lactobacillus and one is B. subtilis. So we will see under the starvation, there is the autoframe rates autoframe genes are much more. So under the low nutrition, so we will see more autoframe rates. So you will see like they are different from the rich conditions, and this is also true in the human cells. For example, in the breast cancer cell lines, when they are deprived of the glutamate for a relatively long time, we will also see there is a massive autoframe happening under these low conditions. And magnetically, we found those high frame shifts are more are also more enriched in those codons, which are non-optimal codons. So then we wondered, so what is the physiological relevance? So when we go back and see what can be the functional consequence of this mechanism, that reminds us of a system which is a toxin and antitoxin system in bacteria. So basically, what is that? It's like under the normal conditions, this toxin and antitoxin system, those two proteins will sequester together. But when there is low nutrition happening, or when there is a stress happening, proteins will degrade one of the antitoxin proteins and release these toxin proteins. That is also kind of RNAs and fast degraded RNA. And this fast degrading RNA can sequester those cells into the persister cells that can allow these cells later to survive better. So we want to see if NMD is also kind of function in the similar ways. So the first thing we check is we compared the cell growth with the strains with NMD and without NMD in the rich condition versus with the low nutrition condition. So we find that cells with the NMD grow slower. So this suggests that NMD can limit cell growth in the low nutritions. And then we want to know if this, we also want to know if this is better, it's beneficial for them after long-term like stress. So then what we did is we measured the cell survivals after we culture them in the long-term stationary stage, and we find that those wild type, so the cells with this NMD, their survival is higher than the one without NMD. So this suggests that this NMD decay, they can function similar to what bacteria had this stringent response. So to summarize, I have shown you that we find this -1 frameshift, it's induced in these low nutrition conditions. And this NMD can actively degrade those the bulk transcriptome in the low nutritions. And we found mechanistically it's low codon optimality instead of this program frameshift are induced the frameshift process. And we also show that this is conserved not only in yeast, but also in humans and also the prokaryotes as well. And in the end, we show that this NMD can limit the cell growth in the low nutrition conditions that can help them better survive after long-term nutrition deprivations. So from that, I would like to thank for my group and all the people in the lab, and also to all the grant and collaborations. Thank you. And also I'm happy to take any questions.
Thank you so much, Yujie. And we can go to questions. I think you explain many interesting things. I'm sure there must be questions. So we open now the microphone for anyone who wants to make a question, to be sent to the first part of the seminar or the second. I can start because I was thinking in your experiments, you use this nutrition deficient media that I guess it lacks or has low levels of many different amino acids. But if
Yes.
you use a media in which you only deplete one or another amino acid, then you see this frame shifting that occurs in higher proportion in the codons encoding for this specific amino acid?
Yes, yeah. So basically, this condition is very poor in all the amino acid. So we haven't tested for dropout for single amino acid, but we tried with one. It's SD, like a different synthetic complete medium, which a little bit higher of all the amino acid, maybe 30% higher of all the amino acid, and we didn't see that.
Because if I understood well, your prediction is that it's like the cis signal is the codon optimality, and because the coding tRNA for this codon is limited, and then if you make like a change in codon, artificial change in codon optimality, let's say to a media in which you don't have one of the amino acids, so you expect this tRNA decoding, it's not charged, and then you expect to see frame shifting like it did to this codon or not?
Yes, that is something we are trying now to either deprived of the specific amino acid in the conditions, or just trying to inhibit some specific tRNA, charged tRNA. Yeah,
And
And
a second question. So, do you think that the signal for the degradation of these mRNAs that they have these ribosomes stalled in this, or they have made, sorry, this frame shifting? So the signal, which is the signal for the degradation through the NMD machinery? Because the ribosomes are out of frame, and they're encounter, like stop codons, or what is the signal?
Yeah. We analyzed the signal. It's the detected of the premature termination codons. Yeah.
Okay.
Yeah, this is extremely frequent. So you did some analysis.
Yeah.
I think it's like in less than 5 to 10 amino acids, you find the stop codon in the genome.
Okay.
It's really frequent.
Yeah.
So then any codon can find them.
I think 98% or 96% of the events will lead to a premature termination codon.
Okay. Okay, thank you. And Vicent, if I can make you a question about the first part. So I think it's such a great, possibility that you develop this test. But I don't know if I understood well, but if you take a sample for patients and then you do this, this is a complex sample. It could be with different bacteria.
Yes.
You use antibodies, and then it's just to check which antibody or which combination of antibodies you can use with this sample. Although if you don't identify the bacteria that they are there, is right or?
So basically we could deconvolute. So it depends where does the sample come. Like if we have a sample that is comes from blood or things like that, hopefully there is not many things growing there. So most of it will be the pathogen. And then we could test if these antibiotics that we're using will work or not. But if we have a complex sample, we did, for example, some experiments with fecal cultures that have hundreds of species.
Okay.
And then we could tell up to what degree each one of the species is responding or not. So we could do kind of a phenotypic. The good thing is that we do the analysis all together, and then computationally we split. Of course, the more species, the lower power, but sequencing is cheaper in comparison. So that's kind of the angle. But that we do it routinely already in the lab. But then for bringing it to the clinic, we want to make it very, very, very easy, because it's a very different standard to have something that is feasible in the clinic and something that is feasible in the lab. So in the lab, we can already do that. We do that already. Complex samples with hundreds of species, we can tell which ones of them are responding or not. But then bringing it to the clinic, we have to make it. We are making it simpler, but let's say there is many bottlenecks that we have to jump and convince.
Okay, thank you.
Thanks.
Anyone who has
There was I think Puri.
Yes, I have a question. Amazing talk. Congratulations to both of you. Am I interested in the mammalian part? Sorry, that I know is not your biggest part, but I was wondering two things. One is, out of all the mRNAs that are frame shifted, what is the proportion? So, is this a super abundant feature? Or is this happening very nicely, but in a low proportion of mRNAs?
You mean the proportion? It's proportion to the highly translated, or just proportion of mRNA?
So out of the ones that they don't have good codons, that you will see the frame shifted many, more recurrently, from the 100% of mRNAs that you have in the cell, out of your favorite ones, how many of them? What is the percentage of them that are frame shifted?
I think we have shown it like in the different cell lines. It's quite high with those undergoing decay, and they are frame shifted. It's like 90%.
So this
something like that.
This is in mammalian cells, right?
Yes, in the breast cancer cell lines undergoing 48 hours of the glutamine deprivation. Yeah, it's from public data. It's not we performed the experiment, but it's from the already published data. We reanalyze them. Yeah.
Yes, just to complement, there is a bioarchive, and hopefully soon the final paper should be published soon, hopefully. But there's a bioarchive where there is this information. But the key point here is the conditions. In very strong deprivation in mammalians, like this 48 hours glutamine deprivation, basically there is more RNAs being frame shifted and non-frame shifted at bulk level. So that's massive. The thing is that probably people is weird, and this is a very strict condition. But we have evidences that this is happening also in patients in clinical samples.
So that was my next question. Are you able to see the frame shifted protein, or is it extremely unstable?
Yeah, it's extremely unstable. Because now we are doing yeast, it's not so much protease inhibitor that can be used, right? But maybe in mammalian you can use protease inhibitor to proteasome inhibitor to get those unstable peptides. Yeah, but it's extremely unstable.
Okay,
and they are very short. Yeah.
Yeah, my last question is, what is the influence of the integrated stress response that you should induce because of amino acid deprivation into this? Is this happening predominantly in things that are being translated under stress, right? You know what I mean?
Yeah. Yeah. I think we are doing also, but also in yeast, we do with the GCN.
Yeah, you will do GCN1 depletion.
Yes.
Yeah. It's yeah. Sorry.
No, so sorry, just use your paper.
No. Yeah. So we do it with the GCN2. Yeah. But yeah, it's quite complicated.
So yeah, just to complement, so even if it happens mainly when there is lower codon optimality, it happens in all the genes, in all the codons, in optimal and non-optimal. It's just happening a bit more in the non-optimal because they are a bit more limiting. But it happens in all the conditions. So that's an important concept. And the other thing is that so if you do a drastic depletion of amino acids, that you will fire this, you don't see this. So we have the experiments that we just put the cells for 1 hour, 2 hours in no amino acids, and then you will fire all these things, but you don't see this. This needs really, really long-term multiple generations. So the cells are exponentially growing in a very, very poor nutrition deficient. So, of course, the other thing is also happening, but it's kind of parallel.
So at the end, my last question, at the end, they will die.
No,
Yeah.
they're just going very slow.
But
Yeah, very slow.
most of the new proteins are being, are not being translated efficiently. We do not have mRNA, so it's amazing that they still survive, right?
Our hypothesis is more that this kind of mechanism makes that when you remove these things, the cells will grow faster. When you remove NMD, the cells grow faster, but then we'll start accumulating many rubbish or problems. So what we think is that this is kind of a way for the cell to sense there is no nutrients, decrease protein production, decrease RNA in the cell, slow down, maybe help to hibernate, go to kind of a persistent mode. And that's more kind of the hypothesis. And that's kind of what we see, that the NMD is slowing down growth, but increasing survival long term.
Yeah.
So we think it's kind of, that's why we have this parallelism with bacteria, that there is this kind of, we think somehow it's kind of helping cells to go into a bit lower metabolism stage. But basically it's kind of a new way for the metabolic information to go back to gene expression, in this case using the ribosomes as a sensor. Kind of our working model.
Okay, very nice. Thanks. I now is coming a question after all this discussion. Under these conditions, the cells prepare for this starvation, increasing the transcription and translation of proteins to survive, like chaperones, let's say. So what happens with the things that they have to be translated with these mRNAs, like a lot, to get chaperones and protective proteins for the stress condition? Then you see that there is also frame shifting, or frame shifting is somehow, limited for these proteins that are needed to survive these conditions. I don't know.
Yeah, we had some geo analysis for those high frame shifted genes versus low frame shifted genes, and I think those like ribosomal proteins, which is more optimal codon, they had less frame shifted. Yes.
And did you look at chaperones, or
not specifically where we enriched any terms related with chaperones? Yeah.
Okay, yes, because you need like lots of these proteins under starvation conditions. So then I was wondering, if you have this frame shifting, then this is not efficient for these proteins that you need. It could be because the reason you said is to slow down, like the growth and everything, metabolism, translation processes. But in this case, anyway, you need some protective proteins. So, okay.
Yeah, we don't know really, but in a way, if you engage NMD, you decrease the RNA, so you decrease the protein production in general.
Yes.
So that will decrease the need that you work, but yeah, I mean, we have in the paper, we have proteomics data of everything, so the information is there, but we just did not focus on that. But yeah.
Okay, thank you. Very interesting. So, do we have more questions?
There's a question in the chat.
Yes.
I don't know if you can see it.
Yeah. So is the frame shift and subsequent mRNA decay process selective for certain mRNA types? Do you want to take it, Yujie? Or
certain mRNA types, like which kind of mRNA, or like?
Yeah. So I imagine it's asking if it's so yes to. So it's not, for example, it's not only happening for stress response genes or Fox-keeping genes. So it's happening to everything, but slightly more for those genes that have non-optimal codons, that are normally stress genes. That is a bit counterintuitive, because those genes are probably the ones you think you will need more in these low conditions. But it's happening to all the genes, like the bulk, like most genes are suffering this. So that's why we think it's a more general break for the whole cell, but it's not that it's affecting much more, or at least we have not been able to identify like I don't know, a specific subset of genes that are especially targeted. Just this general codon. The other thing is the codon optimality seems to have some influence in the degree of frame shifting, but that's it.
And did you check also because in GIST retrotransposition, like TY1, another retrotransposition, sorry, they need to do frame shifting for replication. Did you check if under these conditions that you have more replication, more transposition of TY1 elements?
There is not so much TY1 elements that are in the data. I think I checked one or two, but it's not very much captured in our case.
But we have not sequenced the genome, I think, after these conditions. No.
No.
So.
Okay. So it's not, yeah. We
didn't sequence the genome after this.
Okay.
I have a quick question. Mostly to, well, actually, let's see. The frame shifting, you use this HIV sequence, which is mostly T's. Yeah, so like T. So in a way, it makes sense that it will slip. But why prolines? The codons for prolines? Is there something special with proline codons?
Yes, because in our top, so for example, like when we are talking about the codons, which is it's induce more high frame shift, it's the most cases are the proline or arginine, those codons, like the most non-optimal codons in is like proline and arginine, like CGG or CCG or CGA.
Okay. Is it only when you have two of them next to each other, or even the single ones?
In this reporter system, we cloned 6 of them at the same time.
Okay.
Yeah, like 6.
All right. And connected to this, I have one more question. Did you check if like trans factors, like for example polyamines, are regulating this process? Because it is known that polyamines regulate some frame shifting in some specific genes, in some specific mRNAs. So did you check if there is any role for polyamines or the translation elongation factor EIF5A in this increase in frame shifting under low nutrient conditions?
No. We didn't check that.
Okay.
Because that is more related with EIF5A in the more in the conditional way, right? We didn't specifically look at that.
Okay.
And proline is just one of the things that happen, but yeah, we don't really know, and also in a way it's kind of affecting everything because we tested many things. We tested many stresses, but of course I don't know how many libraries are in this paper, probably like 500 libraries or whatever crazy number. So at some moment we had to stop because otherwise we'll not finish the PhD.
Yes. Yeah.
But yeah, but that's what probably we should or at least consider how we can test it in. And now we have a reporter system, so now it's much easier to screen things.
Yes. Okay, yeah, I'm sure that with all this big amount of data that that you are telling us, then some more investigations can be made from this data. Okay, I think there is something in the chat.
No, I think it's the same one than before.
Okay. So I don't know if we have any other question. Okay, so in case we do not have more questions, then I want to thank you very much for these nice talks. I think it's very interesting, and I'm looking forward to reading the paper that I think it will be complex and for some time reading. So thank you, Vicent Pelechano. Thank you, Yujie Zhang. And so we will, as you know, this is record. So anyone who wants to watch again, to hear again the talk, will be ready in our Translacor webpage soon. So thank you very much for the talks.
Thank you. Bye-bye.
Bye.
Goodbye to everyone.
Bye.
Have a nice day.
Bye.
Good. Bye, Paula. Hasta luego.
Bye, Vicent.